Good day, everyone, and we are down to our retroviruses. So let's start first with the question, what is HIV? HIV, or human immunodeficiency virus, is a virus that attacks the body's immune system. If HIV is not treated, it can lead to AIDS, or acquired immunodeficiency syndrome. So, a fun fact, where did HIV come from? HIV infection in humans came from a type of chimpanzee in Central Africa. Studies show that HIV may have jumped from chimpanzees to humans as far back as the late 1800s. The chimpanzee version of the virus is called simian immunodeficiency virus. It was probably passed to humans when humans hunted these chimpanzees for meat and came in contact with their infected blood. Over decades, HIV slowly spread across Africa and later into other parts of the world. The virus has existed in the United States since at least the mid to late 1970s. As for the content of this topic, so first we have the classification and structures of the virus, pathogenesis, clinical manifestations and complications. We have the laboratory diagnosis and identification, and lastly, treatment, prevention, and control. So let's start first with the composition of our retroviruses. So they are made up of 2% RNA, 60% is made up of protein, and 35% is lipid, and lastly, 3% of their composition is made up of carbohydrates. So for the proteins, so it contains a reverse transcriptase enzyme inside the virion. For their replication, the reverse transcriptase makes DNA copy from the genomic RNA. And for the maturation, the virions bud from plasma membrane. So DNA, such as your provirus, will integrate into the cellular chromosome. So this provirus is a template for the viral RNA. So the outstanding characteristics of our retroviruses is that the infections do not kill the cells. So it may transduce cellular oncogenes or may activate expression of the cell gene. So proviruses remain permanently associated with cells and are frequently no expression. So many members of retroviruses are tumor viruses. Now let's proceed to the structure. So the virus is composed of a capsid core which contains the genetic material that has been surrounded by a protein envelope. So the protein envelope has many spikes of glycoprotein, the outer part of glycoprotein called GP120, in which the function of GP20 is that it determines viral tropism by binding to target cell receptors. So this GP120 is attached to the GP41, in which the function of our GP41, it mediates fusion between viral and cellular membranes. So the binding of GP120 to the target cell and attaching to the GP41 is the inner part of our glycoprotein. So the envelope of HIV also contains other proteins including some HLA antigens or human leukocyte antigen. The genome of HIV contains two helices of RNA molecules in folded form, and then the enzymes reverse transcriptase is present, which is responsible for the conversion of the RNA to form the DNA, and there is another enzyme called integrase which helps the viral genome to incorporate into the host cell. Now let's proceed to the classification of our retroviruses. So we have alpha retrovirus, which contains avian leukosis and sarcoma viruses. Next is the beta retrovirus, which contains mouse mammary tumor virus. And we have gamma retrovirus, which contains mammalian leukemia and sarcoma virus. And we also have the delta virus, which contains or involves the human T lymphotropic viruses and bovine leukemia virus. We also have the epsilon retrovirus, which are fish viruses. We also have your spuma virus, which contains viruses that can cause foamy degeneration of inoculated cell. And lastly, we have the lentivirus. So it encompasses agents able to cause chronic infections with slowly progressive neurologic impairment and including HIV. So for the host origin, so retroviruses are isolated from virtually all vertebrate species. So natural infections are usually limited to a single species. And then all mammalian viruses are more closely related to one another than to those from avian species. Next, we also have exogenous and endogenous classification. When we talk about exogenous, these are spread horizontally and behave as a typical infectious agent. And then usually these are pathogenic retroviruses. Then the next one is the endogenous classification, which are transmitted vertically through the germline. And these viruses are usually non-pathogenic and they cannot transform cells in a cell culture. As for the host range, so we have the presence or the absence of appropriate cell surface receptor. So we have three types. So when we talk about ectopic viruses, so they infect and replicate only in the cells from animals of the original host species. Then the next one is amphotropic viruses. So they exhibit a broad host range. And lastly, the xenotropic viruses. So they replicate in some heterologous or foreign cells but not in cells of the natural host. So many endogenous viruses are classified under xenotropic viruses. As for the pathogenesis, so the pathogenesis of HIV infection is a function of the virus life cycle, host cellular environment, and as well as the quantity of viruses in the infected individual. HIV pathogenesis is basically a competition between the HIV replication and immune responses of the subject or patient via cell-mediated and immune-mediated reactions. So having established that the main target of HIV is activated CD4 plus T lymphocytes through the depletion of the total host cell CD4 plus T cell pool via interactions with CD4 and the chemokine co-receptor CCR5 or CXCR4, the mechanism of pathogenesis still has to be understood, and the pathogenesis results in host immunodeficiency. So we are now on our family retroviruses. So actually there are a lot of genus under the family retroviruses, but as for our topic today, we are going to focus more on the human immunodeficiency virus. So our HTVL1 or our human T-cell lymphotropic virus type 1, they are linked to human tumors, and then less than 5% of the patients has a 5-year survival. And then HTLV-1 can be transmitted via mother-to-child transmission, usually 15 to 25%, and as well as blood transfusion or drug abusers, and as well as sexual intercourse. So we have also this HTLV-1 myelopathy or tropical spastic paraparesis, wherein tropical spastic paraparesis, it is a nervous system degenerative disorder. It is a medical condition wherein it causes weakness, muscle spasms, and sensory disturbance by human T lymphotropic virus, resulting in paraparesis or weakness of the legs. And this HTLV-1 has high affinity to our mature T cells. Moving on to the focus of this discussion, so we are going to have our human immunodeficiency virus 1 and our HIV 2. So they belong also to the lentiviruses. They are enveloped RNA virus, usually 100 nanometers to 120 nanometers in diameter. So HIV infection in humans came from a type of chimpanzee in Central Africa. So study shows that HIV may have jumped from chimpanzees to humans as far back as the late 1800s, as what I have mentioned during the introduction earlier. So it was also mentioned earlier that the chimpanzee version of the virus is called the simian immunodeficiency virus. So it was probably passed to humans when humans hunted these chimpanzees for meat and came in contact with their blood. So the cause of AIDS, cytolytic and non-transforming, is our HIV. So this virus encodes its genetic information in RNA and uses a unique viral enzyme, which is called the reverse transcriptase, to copy its genome into the DNA. So the site of latency for our HIV is our CD4 plus T cells. So we have HIV genome or the genes that is found in the virus. So we have the GAG, the POL, the ENV, and these proteins code for the proteins directly involved in viral replication or our structural proteins. So the next one is our HIV genome or the genes. We are going to discuss them one by one. So the GAG or the GAG encodes for the core proteins, or they are the group specific antigens. And then the PRO which encodes a protease enzyme. So we also have POL. So the POL encodes the reverse transcriptase enzyme or the polymerase. And then the ENV, it encodes the glycoproteins that forms projections. So the gene order in all retroviruses, so we have 5' GAG, then PRO, then POL, and then lastly ENV 3'. And also other proteins present in our HIV, we have the TAT, the REV, the NEF, and then the VIF. So the next, we have the target cells of our HIV infection. So we have HCD4 plus T cells, as mentioned a while ago. And aside from that, we also have our monocytes and macrophages. We have the dendritic cells and our Langerhans cells, the transformed B cells, the astrocytes, oligodendrocytes, and our microglial cells. So as for the transmission, the highest concentration of our HIV is in the semen and blood. So parenteral transmission can also be possible. So we have vertical transmission. When we talk about vertical transmission, so we have congenital transmission wherein the baby can be infected by the virus because the virus can cross the placenta. And we also have the perinatal transmission wherein during birth, the newborn is being infected. And we also have neonatal transmission wherein it can be transmitted via breastfeeding. And then the person who are at high risk for this virus are homosexuals or heterosexuals with multiple sexual partners and then it also includes intravenous drug abusers, and we also have persons receiving multiple blood transfusion, and then the babies of infected mothers via vertical transmission, and lastly, we also have medical and paramedical workers. So those are the persons who are at high risk for our HIV infection. So as for the clinical manifestations, so 4 to 12 weeks is the incubation period for our HIV. And then we have early or acute infection. And then we have high virus production. And then viremia and widespread seeding lymphoid tissues. And we also have non-specific illness. So if the infection is chronic or latent, so patients either may be asymptomatic of developing persistent generalized lymphadenopathy, since the virus can spread to our lymphoid tissues. And we also have the so-called ARC, or the AIDS-related complex, wherein the patient will have unexplained weight loss, the patient may experience fever, oral lesions, and then if ever the patient is on the final or crisis or full-blown AIDS, so there will be a breakdown of the host defense, and then there will be a dramatic increase in viremia. As per CDC guidelines, so any HIV-infected person with fewer than 200 CD4 plus T cells per UL, so it can cause opportunistic infections. And then we also have secondary neoplasms, which is the so known Kaposi sarcoma or Kaposi sarcoma lymphomas. And then we also have neurologic diseases. So only 10% of HIV infected individuals will develop full-blown AIDS after a chronic phase lasting for 7 to 10 years. So most people have flu-like symptoms within 2 to 4 weeks after the infection. So the symptoms may last for a few days or several weeks. Having these symptoms alone doesn't mean you have HIV. So other illnesses can cause similar symptoms. Some people have no symptoms at all. The only way to know if you have HIV is to get tested. But if you will have HIV, some of the common symptoms that will manifest are sore throat, fever, swollen lymph nodes, rashes, muscle aches, fatigue, chills, mouth ulcers, and as well as night sweats. So this is similar to a flu. As for our laboratory diagnosis, so HIV tests are very accurate, but no test can detect the virus immediately after infection. So how soon a test can detect HIV depends on the type of test being used. So ELISA is the screening test for our HIV, and then the confirmatory test is our Western blot or our immunofluorescence assay. But you can also have cell culture, antigen detection, PCR, and as well as antibody detection. So HIV infection is determined by demonstrating HIV-specific antibodies, so on serologic test, which usually appears at some interval of time after the infection. So the polymerase chain reaction detects the HIV genome of the infected cells. So detection of the P24 core antigen in serum also indicates HIV infection. So AIDS is diagnosed mainly on the basis of specific opportunistic infections or cancers coinciding with the T4 cell defect in the absence of other known causes of immunodeficiency. So in addition, HIV infections with neuropsychiatric manifestations or severe wasting are classified as AIDS. But then again, the confirmatory test for our HIV is our Western blot. For the treatment, there is no currently effective cure for HIV. Once people get HIV, they have it for life. But with proper medical care, HIV can be controlled. People with HIV who get effective HIV treatment can live long and then healthy lives and protect their partners. So AZT or azithromycin and other antiviral agents are used for prophylaxis against progression to the disease and for treatment. Opportunistic infections and neoplasms are treated individually. And for our prevention, so today, more tools than ever are available to prevent HIV. So you can use strategies such as abstinence, so meaning not having sex or never sharing needles, and using condoms the right way every time you have sex. You may also be able to take advantage of HIV prevention medicines such as pre-exposure prophylaxis, or it can be acronymed as PrEP, and post-exposure prophylaxis, or acronymed as PEP. If you have HIV, so there are many actions you can take to prevent transmitting HIV to other people. And that would be all for our retroviruses, so mainly focusing on HIV or human immunodeficiency virus. Thank you so much everyone for your time, and thank you so much for listening. Hopefully that you have learned something from this video. Bye, see you soon.